Welcome to the February 2025 LACNETS educational event. I'm Kavya Velagapudi, Executive Director of LACNETS.
I'm Lisa Yen, Director of Programs and Outreach for LACNETS.
And I'm Heather Davis, the Associate Director of Advocacy and Outreach for LACNETS.
Today's presentation is on deciphering genetics and genomics in neuroendocrine cancers. A special thank you to our supporters, Novartis, Ipsen, Crinetics, Curium, Exelixis, and ITM. LACNET stands for Learn, Advocate, Connect, Neuroendocrine Tumor Society. Our name also states the mission of our patient advocacy organization. The LACNET staff includes Lisa Yen and Heather Davis. The board includes Donna Gavin, who is the sister of LACNET's founder, Javana Joy Zambezi, and NET patient Mary Dunleavy, who has been living and thriving with NET for 20 years. We're excited to partner with the Carcinoid Cancer Foundation, or CCF, to provide support line assistance and other programs. For more information, visit lacnets.org/supportline. Now I'll hand it over to Heather.
Thank you, Kavya. Our YouTube channel features over 400 educational wellness videos, testimonials, and podcasts. Click videos or playlists to find patient stories, information on types of treatments such as PRRT, liver-directed therapies, chemo, and immunotherapy. There are videos on imaging, nutrition, guided meditation, our annual conferences, and some of our in-person events as well. And you can also find our videos by topic in our video library on the homepage, as well as the Learn tab. Follow LACNETS on social media. We are on Facebook, X, and Instagram with the handle @LACNETS. And be sure to add us to your Save Sender email list. Now, I'd like to remind our audience that LACNETS webinars are created for educational purposes only and do not substitute for medical advice. The views shared in the webinar are from the experts' experience and insights. Please contact your medical team with questions and concerns about your individual care or treatment. And now I'll hand it over to Lisa.
Thank you, Heather. I've been supporting my husband, who was diagnosed with neuroendocrine cancer in 2015, and together we aim to stay informed and engaged with clinical trials as possible treatment options throughout our 10-year journey. We understand that one of the challenges is finding and understand clinical trials that are right for us. And this is why LACNETS created a clinical trials guide, an easy-to-use educational guide that features several key clinical trials for neuroendocrine cancers. You can browse the site by the type of NET, such as lung, or types of treatments, such as immunotherapy, and you can find brief, patient-friendly videos and information about each trial. We hope that our clinical trials guide can help you better understand clinical trials that might be available for you. I'm pleased to now introduce our speaker for today, Dr. Kimberly Perez. Dr. Perez is a renowned NET expert and a medical oncologist at the Dana Farber Cancer Institute in Boston. She received her undergraduate training and medical doctorate at Georgetown University in Washington, D.C. She completed her residency and fellowship training at Rhode Island Hospital in 2010. After completing her fellowship, she joined the medical faculty at Brown University and served as gastroenterologist at the Comprehensive Cancer Center at Rhode Island Hospital. She then joined faculty at Dana Farber Cancer Institute in 2015. Dr. Perez is a medical oncology physician scientist who provides patient care and conducts clinical research of novel therapies for patients with neuroendocrine cancers. She specializes in hereditary cancer syndromes as well, and she has developed and led clinical trials investigating novel therapies for patients with neuroendocrine tumors. When I spoke to her at a NANETS conference last year, I was thrilled to learn that hereditary syndromes are an area of her expertise, and I invited her to enlighten our community on two topics that are frequently raised and also challenging to understand: genetics and genomics. Thank you so much for joining us today to discuss deciphering genetics and genomics in neuroendocrine cancers. Welcome, Dr. Perez.
Hi, my name is Kim Perez. I'm a medical oncologist at Dana Farber Cancer Institute. I've been invited today to discuss deciphering genetics and genomics in neuroendocrine cancers. These are my disclosures, none of which apply to this discussion. What I'm hoping to do today is to discuss genetics versus genomics, provide a definition, hopefully provide some clarity in regards to the differences, discuss the tests that are used to analyze DNA or genes that direct patient care, also review the common inherited genetic syndromes that have been associated with an inherited risk of neuroendocrine cancers, and then conclude with genetic mutations that are used to direct treatment of neuroendocrine cancer. So let's start with genetics versus genomics. I think the two terms are unfortunately sometimes used synonymously, and I think it's important to get a sense of why they're different. So, genetics truly is the study of how certain traits like color, hair, and in this instance, inherited risk are passed down in the genes from one generation to the next. Genomics is a more all encompassing term, kind of the umbrella term for how genes are expressed and how they interplay. So, how do messages get relayed within the body? So this is just a little diagram to provide a sense of what genetics really means because I'm a visual person, so hopefully this will help you as well. So if you think of genetics, you have on the left of this slide is two biological parents, so a male and female, and they come together and their progeny, their child, has their own DNA set of chromosomes. And within the chromosomes, as you can see, every human has 22 plus their sex chromosomes, and there's pairs or copies of each one. And within those copies, it's kind of a mosaic mix of inherited genes from mom and inherited genes from dad. And from there, it becomes this structure. So, this is kind of looking at that purple box and the pair that is within it. So, one of the pairs is that X shaped figure, which is a chromosome. It lives in the nucleus of the cell, and the cells are the building blocks of the body. If you break down that chromosome in more detail, you can see kind of the strand like effect that's included in this cartoon, which is just to show that it's a huge structure that consists of genes. And within the genes, there are nucleotides. And these nucleotide base pairs are essentially the sequence. And when I tell many people to try to convey what this really means or how the body interprets it, is it's the instruction manual of the body or of the cell. So the pattern changes depending on what gene it's sequenced for. So you can imagine over time when you're reading instruction manuals, sometimes those genes or the sequence within the gene can be read wrong. And so you correct it, and the body has its way of correcting it as well. So that's kind of a summary of genetics. Just to tag on to what I was just mentioning about the instruction manual. So we think of the genes. So we have the chromosome, which is composed of multiple genes. And again, the genes are composed of the sequence of nucleotides. And the nucleotides provide a sequence that basically is a way for the cell to relay information. And so, again, this is where there is a message that's being passed along by the sequence. And so you can see your normal would be in this example is DNA has all you can ask for. And then there's multiple different types of mutations. So mutations just mean that one of these nucleotides or multiple nucleotides are different compared to the quote unquote normal sequence. And so you can have things change, like in the second example, the word you changes to O. And essentially, it's not changing the essence or the meaning of the sentence. It's just a spelling error, right? So that's a silent mutation. Nothing clinically will result likely from that error or that mutation. Or you have missense where the Y in you changes to lose. So obviously, that can have an impact depending on, in this sentence, DNA has all Lou can ask for. That's a very kind of changes the whole meaning or at least the message that's being implied from the sentence. And so that could have an impact on the gene's expression. The next is nonsense, where you have a part of the gene code just is cut out. And so that, as you can imagine, all of a sudden the sentence isn't complete. So, yes, that too can result in the gene not being clearly read or interpreted by the cell. And then you have the frame shifts where, just as it implies, you just have a change around of the letters in the sentence. So the peptides don't change, it's just that they change placement. And so, next, the question that many people ask is, okay, so we have chromosomes, we have genes, there's mutations. What does that have to do with my cancer? And so, or my inherited risk. So, you have what's called germline mutations. And again, that is the changes in the DNA code that are passed from biologic parents to their offspring or their child. Whereas somatic mutations, all cells in the body have that set of chromosomes that I described in the earlier cartoon, and they all have a similar DNA code from cell to cell. And so, what can happen, as I mentioned, the code can get read wrong, maybe in a particular cell. And that's where a mutation can be created if it's not corrected. And so, that's something that happens during the lifetime of the human being versus something that is passed down. And so, it doesn't necessarily, the somatic is a mutation that's not going to be found necessarily in the parent DNA and won't necessarily be passed down to the offspring, whereas the germline is something that's inherited. And because offspring are 50-50 of their parents, there's a 50% chance that they'll pass that same mutation to their offspring or their child. So here we have what happens, or what we have learned is one of the ways that cancer can develop. Again, the cell instruction manual is the DNA, and its function can be influenced if there are mutations in the wrong place. So you imagine if you have a normal cell and within a particular gene, a mutation develops, the cell is able to manage that. But in the pair of the second mutation, if that affects the pair in it, you now no longer have that compensatory or that correction that the normal part of the pair was providing. That can result in changes in how the gene or the DNA is read. A third mutation causes more complexity or more changes in how the DNA is being read, and the function is then being affected. And then more and more mutations, because all of a sudden the repair system isn't working. It didn't catch the first mutation, it becomes a little bit more inefficient with the second. And then by the third and later mutations, it's really just not cleaning up house because it's overwhelmed. There's too many things to correct. And so this is kind of another way of looking at it, where let's say you inherited a risk or you inherited a mutation in one of the pairs of the genes that may not have any impact clinically on the cell and the way that it works, but during the lifetime of the person, if the other pair in the second chromosome also gets mutated, then the compensation of the normal is lost. And all of a sudden, more mutations are going to happen, and you're going to have the potential for a cancer to develop. And in the non hereditary, there's no mutation that's inherited. And it's just over time that a mutation develops and then another mutation develops. And that's where, again, things go awry. And that concept, if people can access back to their high school biology days, is the Knudsen's two-hit hypothesis. So I'm going to jump a little bit and explain a technology that has transformed the field of genetics and genomics, and that's called next generation sequencing. So about 15, 20 years ago, when I started seeing patients with inherited risk, we had the technology to sequence, because of the Cancer Genome Project, the DNA. But because the technology was so new, we only had the ability to look at one gene at a time and sequence it. So read the book on one gene, and that was it. What next generation sequencing has allowed us to take a sample of the DNA and sequence the whole DNA structure, and then identify within it certain genes that we want to look at. So instead of saying, I'm only going to look at one gene, I can now look at upwards of 100 genes, and the computer sequence is going to sequence those particular areas of the DNA and provide information on whether or not there's mutations in the genes or in the code that describes the gene. And that is a tremendous advance because now we're able to, instead of having someone get one test and then get a separate follow-up test, this is all one sample. So we're taking one sample from the tumor and we're able to look at the DNA and look at multiple genes that way. Or we're able to look at the germline, meaning your inherited DNA, your main instruction manual, by taking a sample of blood or taking a saliva sample. Or in the case, and I'll get into this in the next couple of slides, if we want to look at the DNA of the tumor that might be traveling in the blood, we're also able to now do that, and it's called cell-free DNA. And so this is what I was mentioning. So these are other terms that many patients will ask about, and they'll see it now in the lay press or on billboards. It's what's a liquid biopsy? What's a tissue biopsy? And how is that relevant to me? And really, as I mentioned in the last slide, really it's just the source of the information. So a tissue biopsy means that we're putting a needle into a tumor and taking a sample of the actual tumor tissue. And from there, we know the pathologist will confirm that we have tumor tissue. And then from the cells that are in that sample, they can then extract the DNA and we can study it. So there's a lot more certainty about what we're looking at. And we can know before even looking at the, kind of in the initial steps of taking the sample, we can know whether or not we have sufficient sample, whether or not we're going to be able to put it into that next generation sequencing technology and be able to rely on the information that's generated. Liquid biopsy is a little bit different. And this is more of what I would call kind of a fishing expedition, where we're taking a sample of blood, which can be 5 to 10 cc. So it's a really small amount. And the thought is that when a cell dies, all it gets broken down in the body, and the blood takes those waste products and they travel through the system and they get excreted through either the urine or the stool. The idea with liquid biopsy is that if healthy cells are getting broken down and traveling in the blood to get cleared out, so should the cancer cells. And so parts of the cancer cells, meaning the DNA or the proteins produced by the tumor cells, may be in the blood. And so that's where it's kind of the big fish net. By taking a little bit sample of the blood is the hope that you're getting some of that sample to analyze. But you can imagine it's easier because it's just, when you're going for blood work and it can be part of that lab visit with the phlebotomist. But what becomes tricky about it, unlike the tumor tissue biopsy, you can't with 100% certainty know that you kind of fished out what you were looking for. We have the technology to determine, is it a good sample or is it a bad sample? We have the technology to tell you that it is from we are able to confirm that it's tumor DNA. The problem is that you may not have gotten all of the information you needed because you're just dealing with pieces of it and not the actual tumor cell and the whole DNA, or the whole instruction manual. Instead, you're getting pages within the instruction manual and you're hopeful that it's all the pages that you need in order to provide the information that would be helpful for care or treatment or diagnosis. So that's the big difference. But the technology is so detailed that we have reached a point where we do feel confident enough to say, yes, we have enough from the blood, and yes, we are identifying a good sample. But what all clinicians should tell you is that it may not be all the pages in the book, so it may not be as comprehensive as doing a tissue biopsy. And with the liquid biopsy, there's even more to learn from it. And I think this is also where it can be very confusing as a patient or someone navigating the health system is, okay, I understand now tumor, liquid, but now what is cell ctDNA versus cfDNA and what's CTCs? I'm looking at clinical trials online and they're talking about checking me for this or that. And did I have that already? I don't know. So this is really just to break it down and share with you all the differences. And so ctDNA is circulating tumor DNA. Again, pages of the instruction manual. You have some of the fragments of the DNA that are derived from the tumor cells breaking down and they're found in the blood. Cell-free DNA, again, fragments of the tumor DNA and non-tumor DNA. So it's a bigger sweep of the blood, or at least what we're looking to investigate is larger because we're looking at the DNA and we're also potentially looking at proteins and other information that we can learn from that sample. And then circulating tumor cells, that's truly tumor cells. So it's the whole tumor. We're not looking for the DNA, we're not looking for the proteins, we are looking for the tumor cells themselves. And so the technology to identify those three types is very different. And they're all blood tests. They all can provide you some kind of quantitatively, meaning how much information can you assess from this is just, with the circulating tumor cells or the circulating, the cell-free DNA, is there any tumor left in the body, right? Qualitatively, it's going to provide that information we can generate from next generation sequencing, meaning what is the sequence? What is the information being provided on the page that we captured? And I think the disadvantages are what we've already mentioned, and it applies to all three, which is we don't know if we're getting the right scoop of a sample, right? So it's not always that reliable. It's not as reliable as getting a biopsy from the tumor tissue itself. And particularly for neuroendocrine, it's not a tumor that makes or generates or results from a lot of mutations in the DNA code. So we, again, may be missing a lot of information here because it's just not that type of tumor. And so I would say you'll see this. It's commonly used in maybe some of the more common types of cancers and or not even the more types of common cancers, but it's just in cancers where researchers have been able to reliably say if we're taking this sample, it is going to provide adequate information from a diagnosis standpoint, a therapeutic standpoint, or a prognostic standpoint. From a neuroendocrine cancer standpoint, it is just not there yet. We have not fine tuned it enough to say that we are using this in real time. And so I'll move on to germline. And so with germline, as I mentioned, these are the inherited mutations that are passed down from generation to generation. And in the background of this slide is something called a pedigree, which is if you ever meet with a genetic counselor, the boxes signify biologically male and the circles are biologically female or born female. And you can have a biological pair that then has children, and that's how you develop what's called the family tree or the pedigree. And then the genetic counselors or geneticists will apply some of the information about diagnoses and their age on this pedigree for a source of information. And so, with neuroendocrine tumors, and really all tumors, there are different genes that have been identified that, if mutated in the right spot or right spots, can convey an increased lifetime risk of developing a neuroendocrine tumor. For neuroendocrine, this is the pie. So I'd say 80% of patients who are diagnosed with a neuroendocrine tumor, it is truly spontaneous. There are the billion pieces of the puzzle that resulted in the cancer developing, and none of which are specific to the genes that we've identified associated with neuroendocrine inherited risk. Then you have familial, which is the 15%. And that's really an area or a group of patients and families where we're still, as scientists, trying to study, is there more there than we know? And what that just means is the field is expanding, it's growing, and that group of patients may not have a DNA mutation that we are aware of or that we know very well. It just means that there's something, but we have to keep working to figure out what that is. And then the 5% are the inherited syndromes that have been well described and that we test patients for that meet criteria for testing. And I just include some here. And to go into a few of them, this is kind of the main group of inherited syndromes that have been associated with neuroendocrine tumors. And as I mentioned, about 20% are inherited. Really, 5% are associated with some gene that we're aware of at this time. And so you have neurofibromatosis, you have tuberous sclerosis complex, the multiple endocrine neoplasia syndromes, von Hippel-Lindau, and familial paraganglioma syndrome. So the multiple endocrine neoplasia syndromes are probably the inherited syndrome that most patients with neuroendocrine have heard of. And many will ask me if they fall into the category that require testing for it. And I would say they are definitely, the representation is the highest for neuroendocrine patients within inherited syndromes, but definitely not a test that everyone should be considered for. And the reason for it is that there are different types of MEN syndromes, and they're all based on the type of gene that's mutated. So MEN1 is the MEN gene, the MEN2, A and B, is the RET gene, and MEN4 is the CDKN1B gene. And then I share this breakdown just to give you a sense that not every neuroendocrine tumor is associated with these syndromes. So the common neuroendocrine tumors that are associated with MEN1 are midgut or foregut, so lung, gastrointestinal, pancreatic. With MEN2, it's pheochromocytoma, and MEN4, again, midgut, foregut, where there is sometimes a higher incidence of foregut, meaning lung. And so I think it's very important. And the reason why we'll comfortably say, I don't think you meet this criteria, again, is because there are other medical diagnoses that are commonly seen with these syndromes. And if you don't carry a diagnosis of the other medical diagnoses, then the likelihood of you carrying a mutation in one of these genes is near zero. Von Hippel-Lindau is a much more rare inherited syndrome, and there's different types. It's more commonly associated with pheochromocytomas, and there have been reported pancreatic neuroendocrine tumors associated with this syndrome, but you can see it's not all neuroendocrine tumors. And so again, not all patients with neuroendocrine tumors need to be evaluated for this syndrome. And then the hereditary paraganglioma syndrome is a much broader kind of, it can have its own tree because it has many different genes that are associated with risk. And it falls within the neuroendocrine family where 40% are associated with an inherited risk. So that is much different than and associated with a gene that we're aware of. And so unlike the neuroendocrine tumors that are not pheo or paras, the risk is much higher of carrying an inherited risk or mutation in a gene. And then finally is just really the families that I counsel and meet with are the ones that tell me, well, this is so strange. Everyone keeps telling me this disease is so rare, but my cousin has a diagnosis, my mom has a diagnosis, you know, how can, and I've had the testing and the testing's negative. What does that mean for me? And a lot of work has gone into families like that. And what we know is, yeah, none of the genes that we're aware of as of today in 2025 have been associated with this familial group. But it doesn't mean that we'll never find it, right? Because at one time, the other inherited syndromes fell into this larger category. And it wasn't until we identified the genes that were associated with them that they started to get pulled out into their own piece of the pie. And so, what we know for the families where there are multiple family members with neuroendocrine tumor diagnosis is that there is a relative risk increase, meaning that the degree of relationship, meaning if my sibling or brother, sister, if I have the disease, then my sibling may have a higher risk. And in this particular study, they quoted upwards of 13% that a sibling could have develop a neuroendocrine tumor in their lifetime. And a parent may have a 6%, which is strange because it's less than a sibling. So that's where we know there's something more than inherited risk that's happening here, right? And then children is even lower because that's always as parents where we worry, we've been diagnosed, are we going to pass it down? And no, you can see from this study, though, limited, but it's from a state that has just such a wealth of information from inherited risk standpoint, which is Utah, is that there's something there. And so the risk is higher with siblings, but less for children and less for parents. So there's other contributors, and those may be environmental, et cetera, but we just haven't identified them yet. So I include this very busy slide, not maybe to break it up, but maybe just to, um, generate conversation. But I think the next question that um, people ask are just what are the genes or inherited risks that are being described? And this recent work has demonstrated that maybe my pie that I initially showed you can apply to all neuroendocrine tumors, but maybe just like Pheopera, do patients with pancreatic neuroendocrine tumors need to have their own tree or their own full pie? And from the work done by my colleagues, it does suggest that there may be some merit to testing patients with pancreatic neuroendocrine tumors. I wouldn't say that everyone should, but unlike, with my patients with pheochromocytoma and paragangliomas, it's more of a, that's just standard practice. We recommend everyone get genetic testing. I think patients with pancreatic neuroendocrine tumors, if more data like this is generated, it may suggest that we need to. And what I mean by that is these are the pies. So this was a group of patients with pancreatic neuroendocrine tumors, and 33% carried a mutation in a gene that's associated with inherited risk and not necessarily a neuroendocrine syndrome. So we have the BRCA or BRCA, which many are familiar with due to a lot of celebrities with that disease. And it's probably just our most well thought out inherited syndrome. And then there's VHL, like I mentioned, MEN, as I mentioned. MSH2 is something called part of the Lynch syndrome, which is commonly associated with inherited risk for colon and uterine cancer. So many of these have not been described with neuroendocrine tumor risk. So this group of colleagues are just saying, hey, we need to keep thinking about this because if not testing them to find a mutation in a gene that we've associated with neuroendocrine tumor, but really to test them because it may illuminate or it may identify a risk in a syndrome that we wouldn't have otherwise considered based on their personal diagnosis or their family history. And someone who gets diagnosed with a BRCA mutation, their cancer related surveillance or age related cancer surveillance is going to be affected significantly. And that's the important part. Because again, this is all risk, it's not diagnosis. So if we can identify that someone carries a risk, then that means that we can have a positive impact on their cancer related surveillance and potentially cure them before a cancer develops. This is a big gear change, but this is just to give you a glimpse into why we test if not all patients are going to, or a majority of patients are not going to carry a mutation. Why are we looking? And the reason is that we are now developing drugs that can target these genes that are being associated with an inherited risk of developing neuroendocrine tumors. And from there, we can learn so much about future therapeutics. But I think it's important that this study in particular looked at patients who had von Hippel-Lindau or VHL related mutations in their tumor. And this drug, a HIF2 alpha inhibitor, was associated with, and again, I apologize for kind of the maybe overly clinical information here, but on the top in box A, where you have the multiple bars with blue and green, what that's showing is response. So each patient's represented by a bar, and there's the dotted line at -30. So all the patients who responded and met, meaning a decrease in size of their tumor burden greater than 30%, it was, goes beyond that line. And that's significant number of patients that are meeting criteria for response. And then the box underneath that in number C is just how long the response lasted. And so you can see it goes from 0 to 32 months. And a great number of the patients had greater than 20-month responses to this drug. And, we call the one on the top right the spider plot. It's just a confusing way. And I would say the take home is it works. It's a good drug to consider. Unfortunately, it is not the cure that we're looking for patients who have von Hippel-Lindau tumors, but it does have a meaningful impact on the response of the tumors associated with this inherited syndrome. So I think to close out, the last couple of slides I have is just really, should I request germline testing? And this is a really important question to ask. And I think regardless of what I'm going to say in this slide, if you are thinking about it and you think it may apply to you, just ask your provider. And from there, I think they should be able to provide guidance as to whether or not you need to take a day and have another appointment, or if it's just not yet, right? And we're still working on this, so it's not a not forever. It's just a not yet. And so if you have other first degree family members, meaning siblings, children, parents that had a neuroendocrine diagnosis and you also carry a neuroendocrine diagnosis, yes, I think you should ask or request to be referred for genetic testing. If you are someone with a neuroendocrine tumor and have hyperparathyroidism or pituitary tumors, yes, you should discuss that with your doctor and see if they can refer you for testing. If you've had more than one primary neuroendocrine tumor, so you've been told you had a gastric neuroendocrine tumor and now you've recently been diagnosed with a pancreatic neuroendocrine tumor, yes, I think it warrants a conversation. If you've had more than one cancer diagnosis, so neuroendocrine is not your only cancer diagnosis, and you have a diagnosis of colon cancer or breast cancer, it's worth a conversation. The yield may be low, and they may suggest no based on the types of cancers you're describing, because not all inherited risks are associated with all cancers. But I think if you have the question, definitely, if you're thinking about it, then ask your doctor or your clinician about this. If you carry a diagnosis of paraganglioma, pheochromocytoma, this is a resounding yes. Why yes? No reason not to. If your provider has questions, then they definitely need to refer you to a center that has clinicians that can answer this question for you. And then if you have a family member who carries a mutation in a gene that's associated with inherited risk, if they're, a sibling, a child, a parent, definitely yes. If they are your second cousin once removed, no, that's probably not necessary. And definitely not if you've married someone who has an inherited, DNA mutation. No, because you don't share their blood. I've talked a lot about the genes that are inherited, and many patients get kind of confused as to, I have had testing of my tumor and it talks about these different genes. Does that mean I inherited the risk based on that gene? Is that an inherited syndrome? And the answer to that is no. So when we look at the DNA, as I mentioned, of the tumor, we have identified certain genes that are commonly mutated and are contributing to the cancer development process. And so for pancreatic neuroendocrine tumors is probably where we have the most information. But as I mentioned a few slides ago, neuroendocrine tumors just are not they don't carry a lot of mutations in them. We call them kind of bland tumors. But for pancreatic neuroendocrine tumors, MEN1, and that does not mean that it's MEN1 syndrome or inherited risk. It just means that that gene was affected and contributed to the cancer development process. ATRX, DAXX, SETD2, those are all genes that MEN1 can be associated with germline risk, but it can also be something that the tumor acquires a mutation in that over time and it's contributing to the process. Same with the GI net and lung net. But pancreatic, these genes have proven to provide information in regards to kind of confirmation that we have a neuroendocrine tumor. And on top of that, helps us differentiate between NET, meaning neuroendocrine tumor, or grade, in the situation of grade 3, or neuroendocrine carcinoma, meaning the more aggressive active type. So sometimes they look very similar under the microscope, but we can use the molecular features to tell us, is it a NET or a NEC? Because the treatment's going to be different. And so the question from that slide really is, should I request tumor testing? I would say it's a yes, no. I think the larger neuroendocrine centers are offering it kind of as part of care. I think the problem right now is that it's not driving a lot of care options. So the busulfan that I spoke of with the von Hippel-Lindau tumor mutations, that's a very small group. Again, in order to identify that, the only way you can do that is to test. So really, if you're, I think it's again worth a conversation with your provider to make sure that it makes sense. What are we going to do with it? My heart aches for all patients that have testing done with their providers, and it's a yes, no, or they end up with a positive test that doesn't have any clinical significance or relevance, and then they're left thinking, oh, it's a medical test that's positive, so it must be bad, and it's really not bad. So really being thoughtful about testing and the need is important. But as I mentioned, it just can provide us some information about kind of the diagnosis sometimes. And I think right now is probably where it is the most impactful. And so this is really the where I feel like a lot of patients are at this point, because the field has grown tremendously and there's a lot of commercial labs out there. In the bigger cancer centers, we have our own, platforms or next generation sequencing options, and it's just overwhelming. And I have many, patients who come for consultation and they have, reams of paper with all these different, emblems from different companies and different information, and they just, want to make, heads or tails of it. And I would say with anything, any testing, should always feel like you can talk with your providers about it, that there should always be a conversation. And if the answer can't be provided by that person, then redirection to somebody who can is important. Just to conclude, hopefully I was able to share with you kind of the difference between genetics and genomics, who should get testing, what do we know so far about genetics in regards to inherited risk, as well as tumor-related findings for neuroendocrine. And I'll finish here and open up for questions.
Welcome back, Dr. Perez. Thank you so much for such an informative presentation on a difficult to understand topic, two topics. And so I want to just thank you again, and also just let the audience know that this question and answer session is for educational purposes only. It does not substitute for medical advice, so we encourage you to please talk to your medical team with questions or concerns about your individual care or treatment. We have over 36 questions. We'll try to get to as many general questions as possible on this topics of genetics and genomics for neuroendocrine tumors, and that are also in the area of Dr. Perez's expertise. But we may not cover all the questions, and we will not address the questions in the order they were submitted, but instead an order that makes sense. So again, Dr. Perez, thank you so much. And we have, as I mentioned, many questions. So we'll just get to it. And I also wanted to just mention, like, your last few slides were fantastic. You answered a range of questions, especially like, as you mentioned, who should get testing. And so there's some kind of follow-up questions to the who should get testing. So the first one we'll start with is, if I have two relatives, and you touched on this, if I have two relatives, like a parent and uncle, who both passed away of NETs, how can I get more information about possible familial or genetic connection to NET? I think maybe part of this question might have been related to, do I need to find out more about their medical records to see if I need to get tested?
Yeah, thanks, Lisa. So I think what's most important is doing a little bit of in discussion and investigation into your family's history. And so sometimes that's easy and sometimes that's really difficult. So I would first ask the relatives if they have any medical records that indicate what their diagnosis is, just to confirm, and then speak with your care provider and request a consultation with the cancer genetic specialist at your center. If there is not one, then there are many resources in some of the larger neuroendocrine centers that could potentially see you. And if you're in a state that has it, but you're just a distance from there, it's always a great question to ask if they have virtual visit options to discuss these questions.
Yeah. And if the relatives have passed away and there aren't really records available, I don't know how much that matters in terms of, if I'm a relative of them, if I need to know that kind of history from them.
Yeah, so it's not uncommon. Not only is it having relatives who have passed away and you can't answer the question, there's also just the generational gap. So there were a lot of conversations or topics of conversation that just were not shared in prior generations or even in current. It really depends on family dynamics. So as much as you know, and if your concern is present, then meeting with, discussing that with your care provider, or requesting to see a cancer genetics specialist to discuss it is always the best first step. Not everyone who comes in for genetic risk assessment in my clinic gets testing. It's a lot of it is truly just talking about what it's known and determining if there would be benefit.
That conversation is helpful. And this is a interesting question. Is there a link between pancreatic NET and pancreatic adenocarcinoma? I have pancreatic NET, and my father died of adenocarcinoma. His mother died of melanoma. Is this just a coincidence?
So I'd say the answer to that is yes and no. There's no current data that supports a link between the two. But some of the work that has been done in pink neuroendocrine tumor and associations with inherited risk have identified patients or families with inherited risk that may explain the pancreatic adenocarcinoma or the melanoma. And the link to NET is really yet to be identified or determined. So if that is the family history, I think it is worthwhile to sit with a cancer genetics specialist and discuss whether or not testing would be of benefit.
If someone had genetic testing for prostate cancer or another cancer, would the test results help with figuring out pancreatic NET treatments as well?
Yeah, so it depends on when the testing was done, but in the last 10 years, most of the testing has included neuroendocrine related genes. And as mentioned in my talk, we haven't found a treatment that is based on molecular findings specifically. So I think the information from the past germline testing or genetic testing for prostate cancer can be helpful. But right now, if anything, it would probably be in a clinical trial setting, if there is one, to investigate that.
And I know you've already covered this as well. So if there's anything else to add, would it be possible to get genetic testing in relationship to NETs? Genomic testing, sorry, in relationship to NETs.
So yes, so it is right now, because the technology has become so accessible. We are now able to test for NET related genomics, or, as well as kind of the broad berth of all genomics associated with cancer. So if your provider has requested testing of your tumor, it's not necessarily limited to just neuroendocrine. It's kind of everything we know about every gene that we've associated with cancer development. So from that perspective, I think it's what hasn't been the case in the past, probably a decade ago, is that you're getting everything you can, and there's not going to be a reflex test, so to speak, after the genomic testing is performed.
Okay, thank you. And this one is interesting because it has to do with the timing of when to get the testing. Should genetic profiling be done immediately after diagnosis with NETs?
Yes and no. So yes, if the genetic testing is being done to see if there's inherited risk based on the person's personal history and family history, from a genomic testing standpoint, no. There's no treatment that would be delayed in order to get that information. But I would also say no to immediate testing, even with genetic or germline or inherited risk assessment, because it's really, I try to encourage people to think about it as a counseling session. When you've just been diagnosed with cancer, it is very overwhelming. So to add additional testing that may or may not impact your current care plan may or may not be in your best interest at the time. The testing is always going to be available. And I tell people it's really important, as much as you can, kind of sit with your current diagnosis or your most recent diagnosis. And then when you're ready to kind of potentially open that Pandora's box of what might be found from a genetic standpoint, take that next step. Not all people at the time of their diagnosis are really ready to take on another diagnosis.
Wow, that's really helpful and thoughtful as well. I know you mentioned BRCA2 or the BRCA mutations, and this person happened to ask, has anyone looked into whether BRCA2 mutation can lead to NETs?
Yes. So when we do the genomic analyses and do what we call kind of linkage analysis, so we're trying to figure out if the BRCA gene has kind of, when it's affected, is it contributing to the tumor development? We haven't found that connection. We have found that the BRCA gene, which is part of the DNA repair mechanism, there are other genes that have been associated with neuroendocrine tumors, specifically paragangliomas and pheochromocytomas, that are involved in that DNA repair mechanism, but it's not specifically BRCA.
Okay, thank you. I'm going to circle back to your previous comment about when to get the testing. And I think that there's a few questions around that. One was a follow-up related to it. Is it important to know information about your genes in order to determine the source of NET or to know the correct sequencing of treatments?
Yeah, I think that's a great question. And I think it's where the field is really trying to answer that question. One, in regards to sequencing of treatment. Right now, we have many efforts or clinical trial efforts looking at all the really great therapeutic options that are available and when to give what in what order. Right now, it's being driven more by clinical response. And also, colleagues of mine are looking at genetics or genomics of the tumor in order to guide that process. But I'd say we're kind of far from using the information to provide that guidance. I think neuroendocrine tumors tend to be the results tend to be pretty bland, meaning we're not finding a lot of mutations in these genes that we've associated with cancer development and other cancers. And so I'd say the source of NET is still, many different contributors. And I'd say we don't have a good sense of how much any one of these genes that we've associated with neuroendocrine development contributes.
Okay. Are there any of them that would affect sequencing or how you go about thinking about the disease?
I think at this point, the only abnormality or gene mutation that's directing any treatment is the patients with VHL-associated tumors and the drug busulfan. But other than that, I wouldn't say that there are any genes that are kind of pushing us to push PRRT before systemic therapy or targeted therapy before systemic therapy. We just aren't there yet.
Thank you. So on the last slide or second to last slide, you had a number of pictures of different companies. I think that's what that was. This person asked, what genetic test is best, and is one panel better than another? Do they look at all possible genes or just specific to an organ or tissue? And what if NEC is an unknown origin? So many questions here, I know.
Yeah, no. So I would say all tests that are currently commercially available are CLIA approved, meaning there is, a standard process that they're following. So you can be assured that whatever results you're getting from those labs that I had included in that slide, you're getting, a regulated process. A regulated process is in place to ensure you're getting accurate information. So I'd say, at this point, the choice is really clinician dependent and institution dependent. So what organizations are kind of linked and have a system in place to get their tissue and blood to the labs and get results in a quick turnaround time. And again, they all are looking at a very similar panel of genes. So I don't think that any one you're going to get more for your testing than another. And then if NEC is of unknown origin. So, I think I did mention that when we are looking at whether or not we're trying to figure out at this point NEC versus grade 3 well-differentiated net. I think that's probably the one situation where we can really use the genomic information to guide us. But there are probably a few cases where we might find a profile of genes affected that might influence where we think the neck originated from. But I wouldn't say it's we definitely use it, but I wouldn't say that it always provides the answer that we're looking for.
Yeah, thank you. There was actually a separate question as to whether Dana Farber preferred one company or another. And just generally speaking, how do the institutions or the oncologists decide on which company to go with for the test?
Well, at Dana Farber, we, from a genomic tumor testing standpoint, we have our own mechanism or lab within Dana Farber. And I know that other centers like MD Anderson and Sloan Kettering and Johns Hopkins and UCSF, and MGH, they all have their own platform that they use. I know that many of the commercial labs were, started by scientists that participated in, at these larger centers in the development of their platforms and profiles. So I think that's where and again, with the kind of regulation that's in place, I think that kind of regardless of where you're getting your testing, you're getting optimal assessment and reliable data generated. So I wouldn't say that we are favoring one over the other. In regards to liquid biopsy, there's a couple of companies that have just been doing it. Guardant and Signatera, Natera were, some of the first companies to offer it. Does that make them better than the newer companies? No, because again, there's regulation in place so that newer companies that your providers may be sending your testing to, they're all regulated. So, and they provide that all in the final reports about, how they've determined what they're doing and all of that. So I would say, feel comfortable with any of these companies providing you accurate information.
Okay, thank you for that. This is a good question. How often do new mutations get added to genetic testing panels? And would you have to get new genetic testing every time the panel gets updated with new mutations? Or in general, when, if at all, would you repeat the testing?
So I would say that every year there's going to be new genes that are going to be added to the platform. What I would caution people from doing is repeating their testing, every time a gene is added, because many of the genes, our technology is outpacing our clinical information. So a lot of the genes are being added based on, data that's coming in, but the relevance or significance on or impact on clinical care may be lagging behind. So to get updated testing about genes that we're still learning about may be of limited benefit and won't impact your care. And so from a inherited risk standpoint, I typically tell people if we don't identify something on the testing that's available today, come back and see me in 5 years and we can review what's been added and if any of them are relevant to your clinical history. And if there is, then updated testing would happen. But otherwise, I wouldn't be searching that out because the field is growing quickly, but not fast enough to have that as a annual, reassessment.
This is an understandable concern. I'm fearful that the genetic test data may be used by sources external to Dana-Farber or other institutions for illicit profiling purposes. Can any assurance be given to allay my concerns?
Yeah, so there are regulations in place and protections in place for all patients. And before genetic testing is done, our counselors typically go through, kind of guidance in regards to what information is protected, which is all your health information, just like any other, the genetic information isn't different, it doesn't hold different weight to, just your past medical history or your medications, that type of thing. And really nothing can be shared without your consent. So unless you've consented to something, and I can speak from my experience at Dana-Farber, is that we have data registries that patients sign consent to participate in, but any information that is kept or studied is all de-identified, meaning there's not any information of your name, date of birth, or anything like that that would allow someone to be able to kind of trace back any genetic testing results to you. So one, I would say with any commercial lab, your data isn't being shared at Dana Farber's, institutional platform. It's not being shared. And if it is being shared as part of a research protocol, it is your identity is not associated with the information.
Yeah, thank you for that assurance. So going back to your comment about how technology is changing, if someone had a blood test cancer predisposition panel done in the past, so say at Dana Farber several years ago, would that still suffice, or are there newer genetic blood tests for NETs?
So for the genetic or the inherited risk assessment, I would say if it was years ago, like as I mentioned, I usually use 5, and that's kind of a very random number, but 5 years to say, okay, if it was done 5 years ago, then maybe reaching out to the genetic counselor that you met with initially and discussing whether or not an updated testing is indicated would be the first step. But other than that, I wouldn't say that it needs to be done, because I think probably within the last 5 years, we haven't expanded the neuroendocrine profile or platform enough to do repeat testing at this time.
Yeah, thank you for that. I know when my husband was diagnosed and met with a genetic counselor too, she said, check back every 5 years and we'll talk about if you need to be retested. Okay. This is a common question. I know you've talked about this a bit, and a very understandable concern. Should my kids be tested, and when, if at all? Are there any benefits to testing them now versus waiting decades for symptoms versus not at all?
I guess it depends on what testing is being defined as. So testing meaning should children be tested to see if they have neuroendocrine tumors? I would say no. That is all, assessment for tumor of any type should really be based on clinical symptoms and not family history. In regards to inherited risk, if you do have a positive inherited, a mutation in an inherited risk gene, then you and your child should meet with a cancer genetics specialist to talk through if that inherited syndrome is associated with pediatric diseases or diagnoses. And if they are, then getting your children tested would be important. If they are not, then waiting. If children, again, defined as an adult child, again, I wouldn't recommend testing unless you test positive. If you've tested positive for a inherited genetic syndrome.
So if the parent has a known genetic inherited syndrome, then talk to the counselor. Otherwise, you're not just testing kids unless they have symptoms. Yeah.
No,
or family
I mean, there's no indication to test children for genetics unless one of the parents carries an inherited syndrome, because they're 50-50. They're inheriting 50% of the DNA code from each parent. So if neither parent has an inherited risk mutation, there's no way for them to have an inherited risk for neuroendocrine.
Okay, thank you. So speaking of inherited syndromes, what can be done, for example, with MEN1 to prevent passing on genetic disease to an offspring, particularly if the genetic mutation has been identified already in the family?
Yeah, that's a great question. So patients of mine with inherited risk mutations have met with fertility specialists and discussed the option of egg retrieval and testing of the eggs for, testing the DNA of the eggs and then determining whether or not they have it, and then doing in vitro based on that. So it definitely is a process of counseling as well as meeting with the fertility specialist to talk through the different options from that are available.
And again, this is specific to MEN. I heard lipomas and lung nets and pituitary adenomas can run together in MEN1. If a panel was negative for MEN1 years ago, but then these and thyroid cancer run in the family, is there a newer test?
I think there's enough here that if you're asking the question, definitely something to ask and review with your care provider. There's just so many nuances, and when sometimes when people think of family, they think of kind of they're crossing, people who are not blood related, and so we just want to be able to sit with someone, and your primary care provider or your oncologist should be able to do this with you and kind of tease out whether or not meeting with a genetic counselor again would be of benefit.
Thank you. And, just to clarify, what's the difference between a positive MEN genetic test and an MEN genomic test? And what does that mean? Can someone possibly have both? I know you talked about both in your presentation.
So, MEN positive genetic test means that the DNA that you were born with has a mutation in one of the pairs of the MEN gene. Whereas a positive MEN genomic test means that the tumor cell has an MEN gene mutation. So you, as an MEN genetic mutation carrier, your tumor can have a positive MEN genomic test. But you can also be someone who doesn't have MEN and have a positive MEN1 genomic test, because we know that the MEN1 gene, when it's mutated, can be part of the cancer development process. So that is definitely where it can be confusing. And I've had patients ask me when they've looked at their genomic testing, meaning their tumor testing, and seen the MEN and asked if they have the MEN inherited syndrome, and they don't, if their clinical and MEN1 genetic testing was negative.
Yeah. And can someone have both? Is that possible?
It is, and it would be because your DNA that you're born with is still the same DNA that the tumor cell may keep, right? So the tumor cell may have the same MEN1 genetic mutation as the one that you're born with, and that's the situation where both can be positive.
Okay, thanks. Someone actually asked something very similar, as kind of like, it seems like a natural follow-up, but yeah, their pancreatic net had an MEN1 somatic mutation, and I also have a parathyroid adenoma removed and a stable adrenal adenoma, so could I have MEN1?
Yeah, definitely something to talk with your care provider about, because with MEN1, the testing, I think in this situation, it's not having the MEN1 somatic mutation that raises my curiosity about having MEN1, but it's really just having the NET and the parathyroid adenoma and the adrenal adenoma, and talking through with your care provider about whether or not genetic testing should be pursued.
So is anyone studying possible germline genes that may be involved in high-grade neuroendocrine carcinoma, NEC, especially in very early onset cases like 20 to 30-year-olds?
Yeah, a lot is being done. And I think it's hard. What patients have shared with me is just watching what's being published and not seeing a lot being published about their personal experience. But it is happening. It's just a lot. I think we have more questions than answers at this point from that standpoint. And so you just may not see a lot out there, but a lot of work is being done to investigate this.
And I know that you already touched on it in one of your slides about the high grade and how that can help. And this person asked specifically what percentage of NET and NEC patients are based on genetics, and what if the panel is negative? I think, with the high grade, that's a bit of a concern.
Yeah, definitely. I think that the NECs are not typically associated with the inherited syndromes. And so I would say that percentage is low. Whereas with the NET, we know that probably between 10 to 15% of NETs are associated with some inherited risk, and whether or not there's a gene that has been identified that explains it is kind of what we're working on. But most of the time it's limited right now with what we know. So I'd say probably only half of those patients that have an inherited risk, meaning that it looks like something is being passed down from generation to generation, are we actually finding a gene that's explaining it. If the panel test is negative, there are certain syndromes that we have recognized as panel negative but clinical symptoms positive or are associated with the diagnosis. And again, that is a discussion to have with your provider. And if your provider feels like they don't quite have the answer, then meeting with a cancer genetic specialist to talk that through is important.
So there are syndromes that are panel negative but syndrome positive.
Right. So MEN happens to be one of them where you can have, we've been seeing more patients and families where they seem to have a neuroendocrine tumor, parathyroid adenoma or hyperplasia, and a pituitary adenoma. And so that all really fits clinically with an MEN1 diagnosis, but their testing is negative. And what we found is, surveillance and care should be very similar follow-up as patients with MEN1. When those diagnoses are made already, those patients by nature are getting, the type of surveillance that the MEN1 patients would get. So it's just kind of helping patients understand and helping providers understand that we still need to be vigilant about screening for other things that may be associated with MEN.
Yeah, so looking at the whole picture, not just the test. Exactly. Yeah, thank you. This is an interesting question. Can environmental factors like food, stress, and toxin influence the development or progression of hereditary tumors?
So I wish I had an answer to this, and I think many people are looking into it. And probably there's been the most work done in the inherited syndromes associated with breast cancer, like BRCA, or with something called Lynch syndrome that's associated with uterine and colon cancers. And so what has been determined for those cancers, there are probably, some adjustments that can be made, but those adjustments are what we would advise to someone, let's say with Lynch syndrome, who has, we talk about diet high in fiber because it's associated with a high risk for colon cancer, but we also tell that to our patients with colon cancer, or even tell patients that don't have colon cancer. So I wouldn't say that there's something specific per hereditary risk and associated tumor. It's just kind of what we'd apply to any person.
Okay, thanks. And somewhat related, do epigenetics affect neuroendocrine cancer risk?
I think so. We just haven't identified them all yet. I think that there's definitely more here at play than we're aware of, and why the field is not closing the book on this. I think we're still very much trying to develop technologies and use the technologies that are available to sort out what else, right? Because if I'm telling you that your neuroendocrine tumor genomic testing is not likely to identify any mutations, you'll say, well, what's the point in getting that done? Well, the point or participating in data registries or databases is so that we can try to identify, epigenetics is just what about everything else that kind of guides or allows us for the genome or gene to be read and the proteins to be functioning. So I think there's still a lot to be learned.
Yeah, thank you for that. What if someone wants to contribute to research? Are there ways to contribute to medical histories for research purposes to aid in earlier detection in NETs? This person in particular had 2 family members with early NETs, father with a small bowel NETs in the 40s and daughter also small bowel NETs in the 60s.
There are. So, I mean, I only know about my experience at Dana Farber. So, whenever we meet someone in the cancer genetics clinic, or when we meet somebody in the neuroendocrine tumor clinic, we offer them the option to participate in our database so that we can maintain some of maintain the relevant clinical information, so that people like me can ask questions later on about different cohorts. So I would say, if you are a patient and I would ask about the databases that are available at the institution that you're receiving your care. And if you are, planning to undergo any cancer genetic assessment or consultation, also asking about whether or not they have a database or data registry that you can consent to participate in.
Yeah, thank you for that. Yeah, there are ways for sure. And I know that at the NIH, Dr. Del Rivera has a natural history study for neuroendocrine neoplasms as well. So just contributing to research in various ways. So this is a general question. What do the RAND lab tests tell about NETs, and do you use them?
I don't use them as part of my daily practice. I think there's still a lot that we're learning about it. I wouldn't discount it. I just think that, just like every other new technology, there are limitations, and time will provide us, more information about it and the utility.
The second part, I know you've talked about the use of genomic testing on tumors. Is there use for genomic testing on tumors? But the second part of the question, how long are tumors stored after being resected, and are they discarded right after the pathologists are working on them? I think that might be helpful to understand.
Yeah, so at academic centers, tumors are kept forever. We have warehouses of the tumor tissue, and a lot, if someone has consented to be part of a registry, we'll maintain them as long as the registry is fine, is supported and funded through our various research mechanisms. At community hospitals, they don't have the ability sometimes, or the storage space to keep them. So they will have an arbitrary period of time in which they'll determine that they're going to get rid of the specimens that they've maintained. But there's not a set date, because the way that the tumor tissue is maintained, it really kind of freezes it in time, so to speak. One of the ways is paraffin embedded, so it's kind of like a wax preparation, so that the tumor doesn't lose a lot of its features, and we can still look at it years later and investigate and ask questions. It becomes relevant when people have diagnoses of cancers, at a younger age, and I'll meet them, decades later, and in order to kind of put their plan together, I will, if it's available, request it and have the pathologist look at it again and make comparisons.
Yeah, it's helpful to know it's still there, frozen, ready to be used. Okay. You mentioned some of the different testing out there. Is the genetic profiling from circulating tumor cells, the CTC, in the extrapulmonary fluid, urine, and blood usable for classification of cell shape in NET?
Yeah, cytology is tricky. It ends up limiting our ability really to provide probably one of the most helpful pieces of information for neuroendocrine tumors, which is the mitotic index, or the proliferation rate. So I think what the pathologists struggle with is providing a description of cells that are seen in the sample, but will always have a disclaimer about kind of limitations in their ability to provide histologic criteria, which are important, like the mitotic index or proliferation rate.
Great, thank you. Does immunotherapy alter the genome or DNA or cause mutations?
It really shouldn't. So immunotherapy is, when it's in the body, is just trying to manipulate the immune cells to react to the cancer cells in a way that it wasn't doing it before. And so it's not really affecting or manipulating the genome or DNA in any way. Obviously, as immunotherapies have become more readily used, there is always long-term or longitudinal analyses to analyze this, but it's really not one of the medicines that we provide to patients that is getting into the cell and affecting the DNA in that way.
And related to immunotherapy, can you speak to the promise of gene-targeted therapies like DLL3 in NET treatment, including trials, and how to think about treatment sequencing?
Yeah, I think DLL3 is really exciting. I mean, with that approval in lung cancer, there are a lot of companies that are investigating these bispecific drugs that are targeting DLL3. And I think the question that is out there is, can you rely on the presence of DLL3 and the response to treatment? And I think these earlier studies are really going to provide that information. I think what's limited right now is that, just like we do in all cancers, is the companies right now are looking at, the necks with DLL3, and anticipate soon they'll be looking at the, grade 3s with DLL3, or, more well-differentiated tumors with DLL3. But because the lung tumors are where they were originally identified and where the treatment seemed to be having their impact, it's going to move kind of in that direction versus, in the opposite where it's just DLL3 and then that. They're just looking for clinical similarities as well as the DLL3 presence. In regards to treatment sequencing, I think it's any time that it's time to think about a new treatment, you just talk with your provider about what the options are and if there's a trial that may be of benefit to you.
Thank you. It's good to know there's promising treatments and they're more and more personalized. How do you think about germline mutations like TP53 found in NET patients that have low family penetrance of cancer occurrence across the germline overall?
So TP53 germline mutations are associated with what's called a Lee-Fraumeni syndrome. So it's actually a syndrome that puts patients at risk for a lot of different cancers. And I would say NET isn't one of the cancers that we typically associate with Lee-Fraumeni. And maybe that is where this question is stemming from, kind of the low penetrance, meaning the presentation or the association with NET just doesn't seem to be as high as other cancers associated with a TP53 germline mutation. And so I would say that if it's present, I think it's and a neuroendocrine tumor has been diagnosed. I can't say that the current data would necessarily link the two.
Thank you so much. This is a good question. I know some people have had VUS shown on their genetic testing. So this person said, I had genetic testing done that showed a few VUS, including an MSH2 that was classified as likely benign. Can you discuss this based on your pancreatic NET mutation slide? And what does VUS mean?
Yeah. So VUS is a term that means variant of undetermined significance. So if you think of the code, if the DNA code is just trying to convey the message, the woman ran down the street and ran, changes to the woman walked down the street, that change, that mutation that changed it from ran to walk doesn't really change the essence of the sentence. So that's where we can call certain mutations VUSs because they're not changing the function of the protein. And in some cases, the VUSs stay as VUSs until we get a certain amount of data that would support either upgrading it to actually having an effect at some point in the DNA or the cellular process, or it gets downgraded to benign because we see enough patients that are enough VUSs that in that specific location that we can say, okay, it's really not anything. And so when a VUS is found, it's not a diagnosis of disease. It's just okay, there's an abnormality in the sequence and we need to follow it. And for a lot of the commercial companies, what they're doing is with all these VUSs, they're compiling all that information. And so if it ever gets upgraded or downgraded, the patient will always be informed.
Yeah, thank you. I know that can be confusing when you get a report with the VUS on it. So we're going to just take a couple more questions. What can you tell us about MEN1 and DAX mutations in the tumor, especially if it's found together in combination, as far as impacting treatment sequencing?
Yeah, I would say that we're really at this point using it more as an aid in diagnosis for pancreatic neuroendocrine tumors in regards to diff grade 3 versus NEC, and not so much in regards to treatment sequencing. I think the hope and a lot of work that's being done right now is trying to correlate the presence of some of these more common mutations and sensitivity to treatment or benefit to treatment. And at this point, I'm not aware of any direct links that would, if your tumor had both, that one treatment would be favored over another.
Thank you so much. And we'll just end with this last question. What advances in this field are you most looking forward to, or what final words of hope do you have to leave with the neuroendocrine cancer community?
I think the hope here is that I'm being asked to discuss this. I think the awareness is great. I think that the more we talk about it, the more interest the field will grow. And I think that is going to help many patients and families that are affected by this disease, in that it can help with the surveillance and identifying patients who have had inherited risk, but it also, a lot of what we learn from a genome or genetic standpoint drive a lot of the new therapies that come about. And so, the more we want to know and the more interested we are, I think the more funding we're going to get for exciting experiments and clinical trials, and we're going to keep moving this field forward.
Yeah, thank you so much. We had over 50 questions that were submitted overall. You can see there's a lot of interest, and we really appreciate, I know I could say, the way that you break this down. I mean, even though I've been in this field for a while supporting my husband, this is really hard to understand, and there hasn't been a lot that's changed. And I think that you explain things in a really clear, easy to understand way, and we really appreciate all the work you're doing in this field and on behalf of the neuroendocrine cancer community as a whole. Thank you so much.
Oh, well, I thank you. LACNets is wonderful. I haven't met a patient of mine yet who has not, been interested and involved and found it such a great resource. So I'm happy to be a part of this community.
Yeah, and thank you to all the participants for these really great questions, really sophisticated questions, and it really shows a lot of thought and interest on your part, and also just really interested in learning about your disease and growing and wanting what's best for yourself. So thank you for that. And now I'm going to send it back to Heather in the studio to close us out.
Thank you, Dr. Perez, for your informational and educational presentation and for answering our audience's questions on genetics and genomics. We appreciate our supporters, Novartis, Ipsen, Crinetics, Curium, ITM, and Exelixis. Educational programs like today's are also made possible by donors like you. Please consider making a donation today at lacnets.org/donate. And a special thank you to our technical director at TVP Live, Rich Tamayo, for this high-quality broadcast production. Again, today's presentation and Q&A session have been recorded and can be viewed on our website and video library shortly after this broadcast. If you registered for today's webinar, you may receive a survey via email. We would greatly appreciate you completing this survey as it helps us improve our content and with fundraising efforts. Again, follow us on social media to stay up to date on our upcoming educational events. Our handle is @LACNets. And now I'll hand it back over to Lisa.
Thank you, Heather. LACNets is a community of support and resources. We recognize that you are more than your disease, and we aim to support you as a whole person. To this end, we offer many resources and programs. Find out more on our website, lacnets.org. LACNets has a monthly podcast featuring experts who answer the top 10 questions in their field. Go to our LACNets podcast page to check out the episode transcript, resources, and the 10 questions we asked the expert. LACNets offers health and wellness retreats virtually twice a year. Our next virtual retreat will be Saturday, March 15th from 8:30 to 12:15 Pacific. Join us for a time of meditation, art therapy, yoga, and more. Register online at lacnets.org/retreat. The theme of the retreat is Brilliance, letting your inner light shine. You must be registered to attend this event. Thanks again for joining us for today's educational event. Goodbye, and see you at our next event.